So welcome everybody, and thank you everybody. I want to, on behalf of all the researchers, I want to welcome you here, and on behalf of all the researchers, I also want to thank you for inviting us, because this is your meeting. But it's really, in a sense, it's our meeting, and I think that one of the remarkable things about the CNM community has been this real sense of community, and so I want everybody to feel that we're really all in this together. We're a partnership involving the families, our children, our caregivers, physicians, researchers, basic scientists, as well as members of industry who are working to develop treatments and cures. So this is all of us in this together, really, and that's the way we feel, and I hope that you feel the same way. I'm going to try to give a quick overview of the scientific presentations today. Some of this may at times seem a little mind-numbing. You may look at stuff and a couple chemical equations or something and your eyes will glaze over. Try not to get too deep into the weeds. Don't worry about the details. Just go for the big picture. And we'll try to explain things in the way that we'd explain things to our families. But certainly, there are going to be things that you have questions about, so make sure you ask questions. You know, stop us in the hallway, stop us at the coffee pot. We're all here to interact with you. I couldn't do what I do without learning from you guys. And we can all make better progress together if we communicate. So don't be afraid to raise your hand, to ask questions, buttonhole us, pull us aside, and spend as much time as you want asking questions. So to start with, our first session this morning is called Getting Our Bearings. And I just wanted to give you a brief background on what we've known in the history of learning about the centronuclear myopathies. And even though these conditions have been with us ever since the dawn of history, there's something that really has only been recognized very recently, since the mid-1960s. So the very first report, and the coining of the term myotubular myopathy, was actually, I'll make an admission, when I was 5 years old in 1966. And it was a paper in which Alfred Spiro coined the term myotubular myopathy, because when he looked at this boy's muscle under the microscope, he saw very small muscle fibers with a nucleus in the center, which is abnormal for a patient of his patient's age. And these myofibers looked a lot like what you see in an early developing fetus, the so-called myotube stage of differentiation. So that's where that name came from. A couple years later, there was another study that recognized that this was familial, so it was passed on from parent to child. And in this particular family, you can see that it's only boys, the squares, that are fully filled in, and so those were affected boys. And then the circles were the mothers who were carriers with a dot in the middle. And interestingly, a couple of them with this half square filled in here were partially symptomatic. And so actually tomorrow, Sabine de Chastenay from the CMDIR and Casey Genetti from my group are going to tell you a little bit about a study on the carrier moms, on which we're trying to really quantify and understand what degree of affection there is in women who carry the gene for myotubular myopathy. A couple years later, in 1972, there was another study published talking about centronuclear myopathy, the same phenomena of nuclei in the center, but in this case, it was following a different inheritance pattern, and both boys and girls were affected. And so this was considered a related but distinctly different thing. So jumping forward to the modern era, in 1996 was when the gene for myotubular myopathy was cloned. And this is a picture here of Jocelyn Laporte back not long after he cloned the gene, and Jocelyn today. And through a series of genetic approaches, he cloned the gene. This picture here is a representation of that sequence. And this is really the beginning of our understanding of what causes weakness in children with myotubular myopathy. A few years after that, Marc Bethune, working with Pascal Guichenet and a group in Paris, and in collaboration of Jocelyn and my lab and a number of other people, identified a gene for the non-myotubular form of centronuclear myopathy, like that family I showed you a minute ago. And this is a gene called dynamin-2, causing autosomal dominant centronuclear myopathy. Soon after that, Jocelyn's group identified a gene called BIN1, which is responsible for myotubular myopathy. And then a few years after that, ryanodine receptor mutations is a common cause. And then more recently, my lab identified mutations in a gene called Titin, which some of you have, as a cause of centronuclear myopathy, and most recently a gene called SPEG. So this is a diagram designed by Pat and Sarah Foy that I think kind of represents why we're all in this room. You know, each of you talking to each other, you may say, oh, I'm myotubularin, or I'm dynamin, or I'm ryanodine receptor. And these are really all different forms of related but distinct conditions. So in a few minutes, Johan Boehm, who trained with Jocelyn and now has his own independent professorship at Strasbourg in France, is going to tell us about the fruits of all this genetics and how we use that to diagnose these various conditions and discover new genes. Following that, Ernan Gonorowski, who's working with Jim Dowling, and a neurologist in Toronto, is going to tell us about the natural history and the clinical aspects. So at the end of this, maybe is a good time to take questions for that group of three of us, just about the generalities of centronuclear myopathies. After that, in the next session, I'm going to give a quick talk about how we're using zebrafish to develop new therapies for centronuclear myopathy. Ernan is going to tell you why it's important to understand the biology of these things and the chemistry here. So knowing what we now know about what myotubularin does is leading to insights into potential therapy by affecting some of the various enzymes that control our muscle. And then finally, Johan is going to talk about some of the other genes that cause centronuclear myopathy and what we're learning. And the point of this diagram is that all of these genes kind of interact with each other. And so learning about one of them tells us something about the others. And developing a treatment for one of us may tell us something about how to treat some of the others. Then in the next session, I think after the coffee break, Anna Bujbello is going to tell us about gene therapy and give us an introduction to her work, which she has pioneered over the past number of years. Then Casey Childers and David Mack will tell us about a dog colony. This is Nibs, shown here with Allison Fraze. And I want to give a shout out to Allison for having been responsible for letting this all happen by helping to find Nibs and bring her back to us. And Casey and David will tell us about the recent gene therapy progress that's been made there. After that, Anna is going to tell us about how that work in mice and dogs is now being translated into developing what we hope will be the first treatment for humans. And then Suyash Prasad, who's the Chief Medical Officer for Audentes Therapeutics, will tell us about how Audentes is taking all this and developing what we hope will be the first clinical trials. After lunch, Marcus Renaud and Barb Smith will tell us about respiratory studies, and many of you, I think, have been part of their study, and I urge you to talk to them if you have questions about that. And then at dinner time, Matthew Patterson, who is the captain or the CEO and founder of Audentes, will give us a talk about what Audentes is doing and what the landscape looks for developing these treatments. And Matt, I don't know if you're in the room, this is Lord Admiral Nelson. I tried to get a picture of him when he was a captain, so you're the captain of Audentes. So, and then finally we'll end up tomorrow with a series of talks about a variety of different things. Soma Das talking about how we're discovering new genes and diagnosing genes much faster through next generation sequencing, a very powerful method to sequence all of our DNA at once. Mike Goldberg is going to tell us about the ryanodine receptor foundation and ryanodine receptor related myopathies and how they relate. Casey Giannetti from my lab will tell us about the carrier study that I mentioned a few minutes ago. And then in the second half, Sabine de Chastany is going to tell us also about a carrier study and a number of other initiatives that the CMDIR has, followed by Mike Lawler, who, although not as broad a topic, is the only person who had a title that wouldn't fit on one line for me. He's going to tell us about the tissue repository and how he's using pathology to analyze the dog studies. And then we'll end up, very importantly, with Barbara Wubels of Audentes Therapeutics talking about patient advocacy. And this is actually also a critical area, and she'll tell us more about it tomorrow, but basically it's about looking out for you guys, figuring out what you need, and how to get it for you, and making sure that what we're doing is responsive to your needs, and that when we achieve our goals, that it's then available to you in ways that you need. So that's all I have to say. I'm going to turn it over now to Johan, who's going to tell us about the diagnosis and genetics.